Now, related to introduction to DNA, what we are going to look at, first of all, DNA is like a book. This is a book of your cells. In each of your cells, you do have a set of DNAs, and they are all in the nucleus, or the center, I would say, of your cells. Now, what is interesting is that you're having cells, you're all having the same DNA. However, some of your cells are not having the same functions. This is because when we open the book or your DNA book, we will be able, or your cells, they will be able to open a specific book, a specific page, and that page may now allow us to create what we call an mRNA, a messenger RNA. What it does is that mRNA is a copy of one of the sections or one of the sentences of your DNA book. So we open the DNA book. We are going to open a page. We are going to copy few sentences of that page. Now that copy is called mRNA. And what the mRNA does is that it's going to leave the nucleus. It will go to the cytoplasm, and then it will now attach to a ribosome. And then we are going to see different amino acids that will be now put one after another one based on what is written in the mRNA. Very interesting. So now we understand that DNA is like a book, but for years and years, we didn't know what was the structure of the DNA. And when we learned about the structure, we saw that that was a code, a genetic code. It means that a code that would have all of the information that we need in order to run all of our cells in our body. However, when we are having DNA in the brain cells, that DNA is only using the pages or the genes that are used for producing proteins that will allow that specific cell to function. So you have the same DNA in your brain cells, in your, again, intestine cells, in your eye cells, I don't know, retina cells, in, I don't know, in your skin cells. However, all of the genes will not express at the same time. And this is because of what we call a differentiation of the cells. So those DNAs exist, all of the genes exist, but they are suppressed based on the activity of the cells. Few or specific genes are going to be active. Now, what you're going to learn in this chapter is, first of all, to use appropriate terminology related to DNA replication, including polymerase, DNA ligase, helicase, and Okazaki fragments. Here, I'm going to invite you to read the definitions on your own. You can find them in the book. You can also find them here, because I also wrote them in this document. If not, you can find that from any source on internet. So please read those definitions on your own. Then, of course, you can also watch YouTube movies as well. There's so many that are also well done. Then you're going to be able to conduct an investigation to extract DNA from a specimen of plant or animal protein. The one that we already, that I used for your exam is the onion. And here I'm going to invite you to find a movie on, extraction of onion. So go on YouTube, write onion DNA extraction. And then you're going to see hundreds of movies. They're all very similar to each other, where we will show you how we are going to extract DNA from onions. And then, of course, in your exam also, you do have one of your questions where we are asking you to describe or to explain some of the steps in the DNA extraction of the onion. Then you are going to be able to describe some of the historical scientific contributions that have advanced our understanding of molecular genetics. Here, We are talking about the history of the DNA, of course. Now, the tough questions that I'm going to respond now is, what is a gene? A gene is a sequence of the DNA that will be used in order to produce an mRNA, the mRNA then being exported from the nucleus out, and of course it's going to find itself in the cytoplasm. And that mRNA will be now able to be read and to, again, allow the cell to produce proteins depending on the codes that are within the mRNA. The nature of the protein produced will be different because an mRNA can be now read by ribosomes. Now we are going to go over the codons. It means 3 nucleotides, 3 per 3. Now we are going to read that the first 3 nucleotides are telling us that we have to put amino acid A. Then the next three ones are going to ask the ribosome to get the amino acid B. Now you're going to see that we are going to read that mRNA by the ribosome. Now they are going to be codon per codon. And each of the codon is going to push an amino acid to be added into the already made amino acid chain. So now if you have codon 1, 6, 3, 10, 12, you are going to have a protein made of amino acid A, B, A, D, D, Z, for example. So this is now the sequence of the protein that you have, and this sequence is specific to that mRNA. And that mRNA is specific to the sequence of the DNA that we call a gene that has been expressed. Then how can vastly different organisms use the same molecule for passing on genetic information? So here, of course, this is a big topic, I would say. I cannot explain that in just 5 minutes. So I'm going to leave it on a side for now, but we're going to go back to this, again, the discussion. What is the difference between DNA and RNA? This is mainly the structure. DNA is deoxyribose and RNA is ribose. So now we see that the structure of the sugars or the ribose in the DNA and RNA are different. DNA is deoxyribose and RNA is ribose. Then what is the purpose of having DNA and RNA in the same cell? DNA is acting like a book, like a reference, like the repository of all of the genes that we have. Not all of them are being active, and it will depend which gene is or which set of genes are active in which type of cell. And mRNA is the copy, converted or transformed copy of the sequence of a DNA. So those sequences, we call them genes. And when those genes are coding for a protein, so through the mRNA, those proteins are going to be specific to those cells. So if you're having a hair cell, you're going to have, again, genes that are going to code for an mRNA that will allow us to make hair proteins. If you're talking about the nail, you're going to have in the nail cells DNAs that are having genes that will code for an mRNA that will now allow us or the cell to produce proteins that will make your nails. Now you can really expand those different examples, and now you understand the role of the DNA and the RNA. Then how can the genetic information be copied time after time without creating errors? This is because we do have different systems in our cells and our nucleus that will allow us to even correct some genetic errors. If those genetic errors will not be repaired, then the person may again undergo, I would say a lethal issue. People, they may die. Up to 25% of the concepted eggs, they die because they are not compatible with the nature, because of the chromosomic accidents, or because of different types of deletions or different types of mutations that may happen. These are errors. So when we are having, again, a couple conceiving a baby, so at the beginning, of course, for the first 3 months, we may not, I mean, there is almost 25% chance that that conceived egg is not viable. It's not going to be able to live because of different issues. So all of the other eggs that will reach the level of 3 months, and then they are going to mature in order to become, of course, infants. This is because they already passed that cap, I would say, and they are already having a healthy, of course, healthy cells and DNAs. Perfect. So now for the rest, I'm going to ask you, of course, to please read the content. So the heredity is, again, when genes or specific characters like the color of the eyes or the color of the hair, they are going to be transmitted from one generation to another one. Here, we don't talk about this for now, but we can talk about genetics, where we are going to talk about heredity, where based on the genetics, we will see different characters, specific characters are going to be transmitted into the next generations. So here, this is again the structure of the deoxyribose and ribose. We talk about the DNA and the RNA. Here we're talking about different constituents of DNA and RNA. So these are purine and pyrimidine bases, adenine, guanine, thymine, and cytosine. So again, you can please again read those, of course, and of course look into the book chapters as well. So after that, of course, here we're talking about different manipulations and different experimentations that were again showing us about, talking to us about the heredity in different animals, and from there we understood again how again the heredity works. Here we are talking about different experimentations where we did put in place, I would say, or we showed the existence of DNA, and that was by using viruses that we that are we call them bacteriophages. These are viruses that will only attack bacterial cells. So through the indirect experimentations, scientists could explain, or I would say show, that we do have DNA. So we did know about the genetic materials before being able even to isolate and then to study that. And why we study that? That's because by studying the genetic materials, we will be able to look into gene therapies, for example, looking into repairing existing diseases due to the fact that some genes are not working or some genes are mutated, and the proteins that are being made may not be functional optimally. They may not function optimally. This is why we need to understand about the genetic material. So now I'm going to again ask you to please read this chapter as well, the book chapter as well, because we are going also to learn about the structure of the DNA, which is the double helix. And here that was Watson and Crick who again established that theory, and then it has been seen in microscopy, electronic microscopy. So the structure that we are seeing here, the double helix, exists really, and we do know about the components. So these are the bases. These are the pyrimidine and puric bases, deoxyribose or ribose, and this is how again the DNA is being made. Now, when we talk about a gene, we are talking about a sequence of a DNA. So if this is a DNA, we may now have a copy of this one, this 1, 2, 3, 4, 5, 6 different nucleotides. Those are going to be copied, and the copies are going to be mRNA. Now we are going to have an mRNA with all of those, the copy, which is all of those 5 nucleotides. That mRNA is going to go out to the cytoplasm, and then based on the codes that are on the mRNA, we will see different proteins being made, because these are different amino acids that are going to be added to each other. Each mRNA is making, they may make the same proteins. If not, we do have hundreds of different types of genes. They're all going to make us hundreds of mRNAs. Those mRNAs can go to our cytoplasms, and then they will be able to create 100 different proteins that are all used for different cellular functions for our cells. this is what you needed to learn for today. So again, please read the book chapter, look into again the content of this document, and please also watch the YouTube video that I already put on your website, as well as maybe you would need to even learn more. So please get used to the DNA structure, to the mRNA, to the genes, and understand the definitions that we already gave you regarding the DNA